Hello students, welcome to Zoology Classroom. This is Yel Mahesh, Assistant Professor of Zoology from Tara Government College, Sangareddy. In today's class, we are going to discuss about electron transport and oxidative phosphorylation. These two pathways or biochemical pathways come under cellular respiration. Now let us see the preview of cellular respiration. We have already discussed in our previous classes that cellular respiration is divided into several phases. So out of those, first phase is called as glycolysis. In this glycolysis phase, glucose molecules are converted into pyruvate molecules in biochemical reactions. So here glucose is the substrate which gives energy or which yields energy, and that energy is essential for the survival of the cell. And this glycolysis takes place in the cytosol. So in this diagram, we are seeing a mitochondrion. This is the mitochondrion, and outside the mitochondrion, it is the cytosol or cytoplasm. So in all living organisms, this glycolysis takes place in the cytoplasm only. And glycolysis occurs in almost all living organisms without any exception. So in this glycolysis, what are the end products? Substrate is the glucose. End products are pyruvic acid. So this is pyruvic acid. This is one end product. Apart from this pyruvic acid, some electrons are transferred to NADH. Some electrons are transferred to NADH. So NADH also is one end product. And ATP molecules also will be formed in this glycolysis. So ATP production is called as phosphorylation. Why? Because we know that ATP formation will be done when one phosphate group is added to ADP, adenosine diphosphate. So when adenosine diphosphate molecule adds to a phosphate molecule, it becomes adenosine triphosphate, ATP. So here phosphate group is it is being added to the ADP. So this process is called as phosphorylation. The purpose of cellular respiration is to generate ATP. Why? Because ATP is the energy molecule. It gives the energy. It is the form of the energy. So in this ATP only will be utilized by the cell for the energy needs. So ATP formation, here this ATP formation is taking place at substrate level when the glucose is being converted to pyruvic acid. During that phase only, here some ATP molecules are formed. So this ATP formation is considered as substrate level phosphorylation. Likewise, the next steps are pyruvate oxidation, and next third step is citric acid cycle or Krebs cycle. In Krebs cycle also, some molecules of ATP will be formed. So here in this phase also, this ATP formation is considered as substrate level phosphorylation. So let us come back to the glycolysis. So once after the glycolysis is finished, pyruvic acid and NADH are formed, and this pyruvic acid, it is sent into the cytoplasm with some transport protons. Protons importer molecules will be there in the plasma membrane of the mitochondrial membrane. So in this mitochondrial membrane, proton importers are there. With the help of proton importers, proton molecules and these pyruvate molecules are sent into the matrix of the mitochondria. Once they enter into the matrix of mitochondria, pyruvic acid will be converted into acetyl coenzyme A. So this is a second step in the cellular respiration. First step is glycolysis. Second step is pyruvate oxidation. So as a result of pyruvate oxidation, acetyl coenzyme A will be formed. This acetyl coenzyme A enters into the citric acid cycle, and in the citric acid cycle, acetyl coenzyme A will be broken down into carbon dioxide molecules. In this process, some NADH and FADH2 will be formed, as well as some ATP molecules also will be formed in this phase. So in this way, by the end of citric acid cycle, the glucose molecule will be completely broken down into 6 carbon dioxide molecules and 10 NADH molecules and 2 FADH2 molecules. And here in the ATP molecules, coming to ATP molecules account, in glycolysis, net benefit of 2 ATP molecules will be there. And again, here in the citric acid cycle also, a net benefit of 2 ATP molecules will be there. So on the whole, 4 ATP molecules have been generated at the substrate level phosphorylation. So carbon dioxide is a waste product, so it will be sent to the exterior through the respiratory organs. Now coming to the fate of NADH and FADH2. NADH and FADH2. These NADH and FADH2, they are also having a lot of energy in them. They have received the electrons and protons from this pyruvic acid or acetyl-CoA. So they are having a lot of energy that was hidden in the glucose molecule. So the energy that was there in the glucose molecules has been transferred to NADH and FADH2 in the form of hydrogen ions and electrons. So that energy has to be released in the form of ATP. So to release that ATP energy, to release the energy in the form of ATP, the NADH and FADH2, they undergo a process that is called as electron transport. So next phase is electron transport. First phase is glycolysis, second phase is pyruvate oxidation, next third phase is citric acid cycle. So once citric acid cycle or Krebs cycle is finished, the NADH molecules and FADH2 molecules will undergo electron transport chain. So they will transfer the electrons to the electron transport chain. That process is called as fourth step in the cellular respiration, that is electron transport. So once the electron transport is finished, at the end of the electron transport, electrons will be released freely into the matrix. At that time, hydrogen ions and oxygen will react with these free electrons to form water molecule. So, and that finishes the electron transport chain. So once the electron transport chain is finished, intermittently in some phases in the electron transport, oxidative phosphorylation takes place. Oxidative phosphorylation and electron transport and chemiosmosis, these three are not separate ones. They are interrelated and they are interlinked to each other. So that's why we cannot separate them. Oxidative phosphorylation, electron transport, and chemiosmosis, all these are interlinked and all these are coupled together. So by these three phases, oxidative phosphorylation and electron transport, and through chemiosmosis process, finally ATP molecules will be released. So this is called as oxidative phosphorylation. Why? Because in this phase, oxygen will be utilized to reduce the free electrons and hydrogen ions, so that so oxidize the free hydrogen ions and electrons, so that the ATP synthesis is being taken place in the presence of oxygen. So that it is called as oxidative phosphorylation. So two level substrate level phosphorylations are there. One at glycolysis level, another phosphorylation at Krebs cycle level, and the third phosphorylation occurs during the electron transport. This is called as oxidative phosphorylation. Why? Because this phosphorylation takes place in the presence of oxygen. So in this way, in today's class, we are going to discuss about the events that occur in this oxidative phosphorylation and electron transport, and how the chemiosmosis process helps in the formation of ATP molecules. So let us get into this topic. Harvesting of energy from glucose has three stages. So broadly, we can classify the cellular respiration into three phases: glycolysis, citric acid cycle, and oxidative phosphorylation. In the glycolysis, so glucose molecule is broken down into two pyruvate molecules. And in the citric acid cycle, these pyruvate molecules will be completely broken down into carbon dioxide molecules. So that we have seen in the previous classes. And in the oxidative phosphorylation, what happens? Most of the ATP synthesis will take place in this oxidative phosphorylation. That ATP synthesis occurs due to the presence of NADH and FADH2 molecules. These NADH and FADH2 molecules will be formed in glycolysis, Krebs cycle, and pyruvate oxidation processes. So in those three processes, glycolysis, pyruvate oxidation, and citric acid cycle, in these three phases, a lot of NADH molecules and FADH2 molecules will be generated. Those molecules are having the energy from this ATP molecule, energy from the received from the glucose molecule. So that energy will be released in the form of ATP in oxidative phosphorylation process. And this oxidative phosphorylation process is linked to electron transport. So in this electron transport, what happens in this process, ATP will be generated with the help of oxidative phosphorylation. And because it is powered by redox reactions. Here, redox reactions means reduction and oxidation processes. They will be occurring successively or alternatively. So once the reduction reaction will take place, immediately the same molecule undergoes oxidation reaction. So in this way, next molecule, again it undergoes reduction and immediately it undergoes oxidation. So each molecule undergoes reduction reactions and oxidation reactions alternatively. That's why all the reactions that occur in this electron transport are called as redox reactions. Oxidative phosphorylation, it accounts for the most of the energy, ATP energy generation. Almost 90% of ATP molecules will be generated in the cellular respiration, will be generated in oxidative phosphorylation phase only. So a smaller amount of ATP also will be formed in glycolysis and citric acid cycle that we have already discussed. That is called as substrate level phosphorylation. Only 4 molecules of ATP will be generated in this substrate level of phosphorylation in these two phases, that is glycolysis and citric acid cycle or Krebs cycle. And for each molecule of glucose degraded to carbon dioxide and water by respiration, the cell makes up to 32 molecules of ATP. So almost 32 molecules of ATP can be synthesized from each molecule in the cellular respiration when it is completely broken down. So out of these 32 molecules, only 4 molecules will be generated in substrate level phosphorylation. Remaining 28 molecules will be generated in the cellular respiration process, in the oxidative phosphorylation process. So, and during oxidative phosphorylation, what happens? Chemiosmosis takes place. This chemiosmosis couples the electron transport to ATP synthesis. So, chemiosmosis is a linking process that couples the electron transport to ATP synthesis. So, immediately after the glycolysis and the citric acid cycle, what happens? NADH and FADH2 will be formed in these two phases, glycolysis, Krebs cycle, and also in pyruvate oxidation processes. So these NADH and FADH2, they account for the most of the energy extracted from the food. So food here means glucose, be it glucose or any other molecule, whatever molecule it may be. The energy coming from the food particles will be transferred to NADH and FADH2. So these are actually called as electron carriers because they receive hydrogen ions and electrons from the food molecule. So they are carrying the electrons and protons, so that they are called as electron carriers. So these electron carriers, NADH and FADH2, they will donate electrons to the electron transport chain. And this electron transport chain, upon receiving these electrons from these NADH and FADH2, it powers the ATP synthesis via oxidative phosphorylation. When oxygen is there, and when these electron transport chain receive electrons from this NADH and FADH2, so when these two conditions are fulfilled, that means oxygen presence and receiving electrons from NADH and FADH2. So when these two conditions are fulfilled, this electron transport chain, what happens, what it will do, it will power the synthesis of ATP through a process called as oxidative phosphorylation. So the pathway of electron transport chain is in the inner membrane of the mitochondria. So inner membrane is also called as cristae. We know that mitochondria is having two membranes. One is outer membrane and second one is inner membrane. And inner membrane is having folded structures. These folded structures are called as cristae. So these electron transport chain molecules are present in the cristae of the mitochondria. And most of the chain's components are proteins. So most of the electron transport chain components are proteins and which exist in multi-protein complexes. So all these proteins along with some other chemicals or some other molecules, they are formed in the form of complexes. So they are called as multi-protein complexes. So the carriers alternate reduced and oxidized states as they accept and donate electrons. So as we have already discussed, these electron transport molecules or these electron transport chain proteins, they will receive electrons from the NADH and FADH2. So when they accept the electrons from the NADH to FADH2, that is called as they are reduced. Immediately they will donate the received electrons to the next proteins which are present in the electron transport chain. So immediately when they donate the electrons, then again they will be oxidized. So when the first molecule is oxidized, the second molecule, what it is doing, it is receiving the electrons from the first protein molecule in the electron transport chain. That means now the second molecule is reduced. Immediately the second molecule will transfer or will donate the electrons received from the first protein. So it will donate these proteins to the third protein. So when it donates electrons to the third protein, the second one is oxidized. At the same time, the third one is reduced. So like this, these will be receiving electrons continuously from the NADH and FADH2, and continuously they will be receiving and they will be donating to the next molecule in the electron transport chain. So like this way, all the molecules, they are continuously and they are alternately, they are undergoing reduction reactions and oxidation reactions. That's why all these reactions are called as redox reactions. So these reduction reactions occur in this electron transport chain over the electron transport chain. So this is called as electron transport pathway. So in this electron transport pathway, nothing is happening, but the electrons will be received from the NADH and FADH2. And these NADH, FADH2, they will transfer or they will donate the electrons to the electron transport chain. And this electron transport chain, it is containing a lot of protein complexes. These protein complexes, they will transfer the or they will transport the electrons from this NADH and FADH2. And finally, when the electrons reach the last protein in this electron transport chain, multi-protein complexes, the electron will be released from that last protein in the electron transport chain. At the same time, that electron drops in free energy as they go down in the chain, and finally they will pass to oxygen and hydrogen ions. So when these electrons are transferred to oxygen and hydrogen ions, they will form water molecule. So here in this slide, we can see that electron transport chain. So as we have already said, electron transport chain, it is having a lot of proteins. So and all these proteins and some cytochrome molecules also will be there. All these are formed into a chain-like thing. So it is called as electron transport chain, and they are formed into some complexes. Majorly 4 complexes are there. So this is electron transport chain complex 1. This is electron transport chain complex 2, and this is electron transport chain complex 3, and the last one is electron transport chain complex 4. So 4 complexes are there. And these 4 complexes, they are linked with some other cytochromes also. So here the electron transport chain is having FMN and ferrous sulfur proteins. FMN and ferrous sulfur protein, these 2 are present in the electron transport chain complex 1. And in electron transport chain complex 2, they are having FAD and ferrous sulfur proteins. And in the electron transport chain, they are having cytochrome B, ferrous sulfur proteins, and cytochrome C1. And in electron transport chain 4, they are having cytochrome A and cytochrome A3. Cytochrome A and cytochrome A3, these are the molecules present in the electron transport chain complex 4. So here in this connecting these electron transport chains with each other, there are some linking proteins also here. So this is ubiquinone. So ubiquinone will be linking protein complex 1 and protein complex 2 with complex 3. So ubiquinone is connecting these 2, complex 1 and complex 2 with ubiquinone 3. And ubiquinone is connecting these two, complex 1 and complex 2 with complex 3. And the complex 3 is linked to complex 4, cytochrome C. So this is the structure of electron transport chain. Here, what's happening in the electron transport chain during the electrons are transported over this chain. So NADH, it has received 2 electrons and hydrogen ions. So these hydrogen ions will be released into the they will be released into the matrix, and electrons will be transferred to FMN. So that now FMN is reduced. Immediately this FMN releases the electrons to the FeS2, ferrous sulfur protein. So now it is oxidized, this is got reduced. So now next FeS sulfur protein, it transfers the electrons to the ubiquinone. So now it is oxidized and ubiquinone is reduced. Next immediately ubiquinone transfers them to cytochrome C. So in this way, the electrons will follow this pathway: cytochrome B2 FeS proteins, next cytochrome C1, next cytochrome C, next cytochrome A, next cytochrome A3, and finally from cytochrome A3, the 2 electrons will be released into the matrix. When they are released into the matrix, these electrons will react with hydrogen ions and oxygen molecule to form water. So like this way, NADH releases 2 electrons. They will transfer the electrons to this cytochrome electron transport chain complex 1. FADH2 releases the electrons to the multi-protein complex or electron transport chain complex 2. So FADH2 transfers electrons to cytochrome FAD, and from FAD they will go to FES, and from there they will go to Q and or ubiquinone. Ubiquinone to they will go to cytochrome A, and from here this is the common path. So till ubiquinone, the NADH pathway and FADH pathway both are separate, but from ubiquinone onwards, the remaining pathway is common for the electrons coming from NADH and FADH2. So in this way, from these both molecules, 2 electrons will be transported over these electron transport chain complexes. Finally, when they release into the matrix from the last multi-protein complex molecule, they will join with water molecule, hydrogen molecule, hydrogen ions and oxygen molecule to produce water. So this is the electron transport chain, electron transport process over the electron transport chain. So electrons are transferred from NADH and FADH2 to the electron transport chain. So and electrons finally they are passed to oxygen and hydrogen ions. So the electron transport chain generates no ATP directly. So this electron transport directly, it is not generating ATP, just it is passing the electrons to the next molecules. So it breaks the large free energy drop from the food to oxygen into smaller steps that release energy in manageable amounts. So chemiosmosis is the process by which these ATP molecules are generated in this electron transport and oxidative phosphorylation processes. And the chemiosmosis is an energy coupling mechanism. So electron transfer in the electron transport chain causes proteins to pump hydrogen ions from the mitochondrial matrix to the intermembrane space. So we know that in the matrix, in the phases of glycolysis, Krebs cycle, what happens? A lot of hydrogen ions are also released. So these hydrogen ions they will be pumped into the intermitochondrial membrane space or outer chamber. So they will be transferred into the outer chamber. When this electron transfer is taking place, that energy pumps the hydrogen ions from mitochondrial matrix to the outer chamber or intermembrane space. So later what happens, hydrogen ion molecules will be accumulated in high quantities in the outer chamber or intermembrane space. So when their concentration is increased in the outer chamber, what happens? Across the concentration gradient, these hydrogen ions they will flow into the mitochondrial matrix again, and this flow will be through the what happens through the ATP synthase. So ATP synthase molecule pumps the hydrogen ions from the intermembrane space or outer chamber to the matrix. So when these protons are pumping or flowing from outer chamber to inner chamber, the energy will be released, and that released energy causes the ATP synthesis. So the ATP synthase enzyme, the ATP synthase molecule is using the exorganic flow of hydrogen ions energy. So when hydrogen ions are flowing from higher concentration area to lower concentration area, that results in the release of energy. We know that when water is flowing from high altitudes to low altitudes, automatically the energy present in the water molecules will be released. So that results in the flow of the water. So in the same way, what happens, ATP synthase also uses the energy from the hydrogen ions flow to generate the ATP. So this is called as phosphorylation, and this phosphorylation will be done when oxygen is present. So that's why it is called as oxidative phosphorylation. So here osmosis also taking place, and this is called as chemiosmosis. So hydrogen ions are they are pumped across their concentration gradient. So this process is called as chemiosmosis. So this is the what ATP synthase molecule. So this ATP synthase molecule is having two parts. One is the major part, another one is a stator part. So this is a rotor. It will be rotated when hydrogen ions are flowing through it, and the starter helps in this rotation of this thing. So this is the outer membrane, or in intermembrane space or outer chamber, and this is the mitochondrial matrix. So during the electron transport chain, hydrogen ions will be pumped from this mitochondrial matrix into this outer chamber. So when they are accumulated in high quantities in this outer chamber, the electrons will be pumped into the matrix through this ATP synthase molecule. So when it is pumping through this ATP synthase molecule, what happens? This major rotor will be rotated. The rotated energy will be used for the coupling of inorganic phosphate to the ADP. So when inorganic phosphate couples to the ATP, by utilizing this rotor energy released from this ATP synthase molecule, ATP will be formed. So in this way, hydrogen ions will be pumped into the inner chamber or matrix when they are flowing into this inner chamber from the outer chamber. The energy that is released from them is utilized for the rotation of this ATP synthase. The rotational energy, that's kinetic energy, helps to form the ATP from ADP and inorganic phosphate. So in this way, in this slide again, we can see that this is enzyme complex 1, or electron transport complex 1, electron transport complex 2, electron transport complex 3, and electron transport complex 4. So 4 electron transport complexes are there. So, and when NADH is transferring electrons to this, here at this junction, one hydrogen ion will be shunted into this outer chamber from inner chamber. And again, at this level, during the electron transport over this third complex, one more hydrogen ion will be shunted into this thing. And again, at the fourth complex level, also hydrogen ions will be shunted into this thing. So, and all these hydrogen molecules, they will be accumulated in this outer chamber. And when they are accumulated in high quantity, again, they will be shunted into this inner chamber through this ATP synthase molecule. So when they are coming into this ATP molecule, the flow of hydrogen molecules energy, hydrogen ions energy will be utilized for ATP synthesis. So this process is called as chemiosmosis. And both this electron transport chain and chemiosmosis processes, they can be collectively called as oxidative phosphorylation process. So in this way, oxidative phosphorylation can be divided into two phases. One is electron transport chain, and another one is chemiosmosis. So the energy stored in hydrogen ions gradient across the membrane couples the redox reactions of electron transport chain to ATP synthesis. The hydrogen gradient is referred to as proton motive force. So this force emphasizes its capacity to do the work. So now account of the ATP production by cellular respiration. So during cellular respiration, most of energy follows in this sequence. So glucose to NADH will be formed, and FADH2 also will be formed. The NADH and FADH2, they will transfer the electrons to the electron transport chain, and protons also will be there. So proton motive force will be there by electron transport chain. So the electron transport, when the electrons are transported by this chain, protons will be motivated and they will get force. That force will be utilized in synthesizing ATP. So about 34% of energy in glucose molecule is transferred to ATP during cellular respiration, making about 32 ATP. So from single molecule of glucose, 32 ATP will be formed. This 32 ATP molecules are equivalent to 34% of the energy present in the glucose. So the energy present in the glucose is not completely released. Only 34% of energy will be released in 32 ATP molecules form. The remaining energy will be there in the carbon dioxide molecules only. So what we have to remember in this context is that in the universe, no way the energy cannot be released completely from the molecule. If the molecule is supposed to release the complete energy that is present in this in that molecule, that is not possible. In even atom bomb molecules also, 100% energy will not be released from those molecules when they are exploded. So this is the overview of the cellular respiration. So in the glycolysis phase, 2 ATP net ATP molecules benefit is there, and 2 NADH are formed. And in the pyruvate oxidation, 2 NADH are formed. And in the electron transfer, in the Krebs cycle phase, 2 ATP were formed, and total 6 NADH and 2 FADH2 were formed. So completely, totally 10 NADH and 2 FADH2 are formed, and 2 plus 2, 4 ATP molecules are formed. So here in this oxidative phosphorylation phase, via electron transport and CHEMIOSMOSIS phases, around 26 to 28 molecules of ATP will be generated. So on the whole, around 30 or 32 molecules of ATP will be released from each glucose molecule. So this is the energy budget of this cellular respiration. So with this, we have finished our topic of cellular respiration. In next class, we will meet with another topic. Till then,